Okay, so congratulations. We are over and done with the cell. No more cell. So now we are up into the tissue. So now let's talk about the human tissue. There are 50 trillion cells of 200 different cell types, but they could all be categorized into four different types of tissues, as we mentioned. The first one is the epithelial tissue, and that is the tissue that lines your external environment and the deep lining of your gastrointestinal tract, your alimentary tract, as well as your respiratory tract. There's the connective tissue that connects the basement membrane and connects tissues to other tissues. And then there's the excitable nervous tissue and the muscular tissue. The organ is a structure with discrete boundaries that is composed of two or more tissue types. And with the discovery of the microscope, we started an entire new field known as microscopic anatomy. And that is the study of tissues and how they are arranged into organs. The tissue is a group of similar cells and cell products working together to form a specific role in an organ. They work together to perform a specific function. And the four primary tissues, as we said, are the epithelial tissue, the connective tissue, the nervous tissue, and the muscular tissue. And they differ from each other in the types and functions of their cells and the characteristics of the matrix, extracellular material, and the relative amount of space occupied by the cells in that matrix. The matrix extracellular material is composed of the fibrous proteins, this clear gel known as the ground substance that is found in the matrix of the extracellular material, also known as the tissue fluid or the extracellular fluid or interstitial fluid. So interstitial fluid is kind of an intermediary between the intracellular compartment, that's inside the cell. The interstitial fluid is in the middle between the cell compartment and the extracellular compartment. But both of them, they are outside the cell. They are not intracellular. And this matrix, this extracellular material, this extracellular matrix contains water, gases, minerals, nutrients, waste, and hormones. The human development begins with a fertilized egg, an ovum that united with a sperm, which divides and produces first tissue layers. So the three primary germ layers are the ectoderm. This is the very outside, and that gives rise to the epidermis and your nervous system. So a question on the exam would be, to which of the primary germ layers does the epidermis and the nervous system belong to? And the good answer to that would be the ectoderm. The endoderm is the very inside, and that gives rise to the mucous membrane lining the alimentary tract of your digestive system and your respiratory tract, and as well as the glands. So every gland, the pancreas, the thyroid gland, the no matter what gland are we talking about, that is definitely made up of epithelial tissue of the endoderm, the endoderm. Then between the endoderm and the ectoderm is your mesoderm. So the mesoderm is the middle layer, and it becomes gelatinous tissue called the mesenchyme. So it changes into a phase of the cell tissue structure from the mesoderm into the mesenchyme, and it is made up of collagen fibers and fibroblasts that are in a colloid, a gel matrix. And this mesenchyme gives rise to your connective tissue, and that's your blood and your cartilage and your bones. So understanding histology requires awareness of how specimens are prepared. So histologists use histological sections. These are tissue slides into thin sections, 1 or 2 cells thick. So there is a fixative histological section, and that prevents the decay. So we fix formalin and prevent the putrefaction of the histological sample. And there's staining, histological staining, and that colors the tissue and it binds to different cellular components, eliciting different tissue colors upon binding. The sectioning reduces the three-dimensional structure to two-dimensional slices. There's a longitudinal section and the tissue cut on its long axis, and there is a cross section or transverse section. And here, the tissue is not cut on its long axis as in the longitudinal section. The tissue is cut perpendicular to the long axis of the organ. There's an oblique section, and that is tissue cut at an angle between the cross and the longitudinal section. I did not ask you about that on the exam at all, but this is just to show you that you can never examine a tissue from one perspective and see all about it. You can never have one dimension. So for example, this figure here, what does it show you? It shows you an egg. If you look at it from this perspective, you can only see the egg from outside. You will never know that there is yolk inside, yellow yolk. So we have to have these different sectioning. So why do we have different histological sections? So they can allow us multi-dimensional ability to examine the tissue and to examine and analyze the structure from different perspectives so we can understand the function properly. And this here represents the longitudinal section, and we use that a lot in anatomy. So now when I slice the bone in half, I was able to see the bone marrow inside, and that for me is my integration center, and that is where I receive stimuli in order to generate. It's an effector that mediates the effect of making more red blood cells, making more white blood cells when we are under infection. And this is a cross section that shows me the diameter. Now, and this is an oblique section where you see in the, so for example, it's very easy to distinguish between the longitudinal section, the oblique section, and horizontal section, because longitudinal section, we expose longitudinally. It's very big, but in the cross section, we just look like almost a circle. That's a cross section. But an oblique section, you would see that it is more long than it is wide. It's more long than it is wide. So not all tissues are sectioned. Some tissues, when we are interpreting the tissue sections, some are smeared. So a smear is tissue that is rubbed or spread around a slide. Examples would be blood and soft tissue, like the spinal cord. So we do a blood smear to see the shape of our red blood cells. Are they sickled? Are they normal? Are they hemolyzed? Okay. We do a blood smear to see, is there any microbial infections on my blood smear? Is there any parasite, malaria? Is there any form of infection? And there's also a spread. A spread is different from a smear. A spread is when the tissue is not rubbed. The tissue is laid out on a slide. So it's not rubbed or spread. It's just placed in there. And an example would be the membranes and the cobwebby tissue like areolar tissue. So now let's talk about four different tissues. There's the epithelial tissue, and the epithelial tissue is a sheet of closely adhering cells. Closely adhering cells, one or more cells thick with upper surface exposed to the environment or the internal space. The epithelial tissue covers the body surfaces and lines the body cavities. So it's from the outside and the inside. So if you are facing the external environment from the outside, it is your epithelial tissue that is in approximation, that is in proximity with the external environment. But from inside, so we are hollow from outside and hollow from inside. So the food that goes into our digestive system and it never got absorbed, that food has never entered our body, even though it entered our mouth. It entered our throat. The food entered our stomach, but it didn't get to the body. So it's just as equally as being outside in a shelf, because you are only inside if you make it to the blood. So the epithelial tissue is what mediates the passage from the external environment to the internal environment. And this epithelial tissue is avascular. It does not have blood vessels, usually noticed by the underlying connective tissue. So the underlying connective tissue is the one that receives on behalf of this epithelial tissue all the nutrients and the oxygen. What are the functions of the epithelial tissue? That's something very important to have in mind. Number one, the epithelial tissue is part of our innate immunity. That is our natural immunity. Our innate immunity is comprised of our barrier, of this epithelial barrier, like a fence. This epithelial tissue, it protects the deeper tissues from injury. Also, the bone tissue, for example, although it's connective, that bone tissue is also for protection because the bone is right around the brain. So, you put money inside the safe. The safe is made of iron. It's very thick and strong, and you put the money inside it. Same thing, your brain is more precious than diamond and silver and all the money. That's why it's covered by this safe known as the bone. Your heart also, your lungs, they are very precious. So they are encapsulated and covered from the outside with this bone, bone tissue. The protection protect the deeper tissues from injury and infection. So the epithelial tissue is also important for secretion. So it produces and releases mucus, sweat, enzymes, hormones, and other substances. Epithelial tissue is also important for the excretion, voiding of the waste from the tissue. And for reabsorption, we absorb chemicals such as nutrients from our digestive system into the bloodstream through the epithelial cell. Just remember that these secretions, the mucus, the sweat, the enzymes, the hormones, they are all not secreted by nerve tissue or muscular tissue or connective tissue. It is secreted by epithelial tissue. But what kind of tissue support the epithelial tissue? Connective tissue. Excellent. Very good. Very good. Good job. We just said that. So the connective tissue, that is the one that is working really closely. That's the one that's really helped the epithelial tissue. It is really close with the epithelial tissue. Then the epithelial tissue is also important for filtration, where all substances leaving the body are selectively filtered by an epithelium. The epithelium is also important for sensation, and the nerve endings in the epithelia detect stimuli. What is the general structure of the epithelia? The cells close together, the cells of the epithelial tissue are very tight and close together. They are joined by CAM, C-A-M, cell adhesion molecules. And a small amount of the extracellular material, and it is avascular, has no blood vessels, but it is nourished by the underlying connective tissue, and there is a high rate of mitosis in cells near the connective tissue. So there is a large component of the epithelial tissue, but the part of the epithelial tissue that is closest to the connective tissue, that site undergoes a lot of division, a lot of cell division cycle. That is known as the site near the connective tissue, where there's a lot of mitosis. And the epithelial tissue rests on the basement membrane. Epithelial tissue is resting on the basement membrane, which is a layer between an epithelium and the underlying connective tissue. It's very important to realize that this basement membrane is the layer separating or maybe joining for me on the top your epithelial tissue and connecting it to the bottom, the connective tissue. Collagen, glycoproteins, or other protein carbohydrate complexes and it anchors the epithelium to the connective tissue below it. It's made up of collagen, glycoprotein, and protein carbohydrate complexes. What is the general structure of the epithelium? So the surfaces of the epithelial cells are the basal surface, which is the surface facing the basement membrane. So the epithelia has two surfaces, a basal surface that is facing the basement membrane, and an apical surface that faces away from the basement membrane. And there's a lateral surface that is between the basal and the apical surface, known as the sidewall. The epithelia are classified by the number of the cell layers. So there are simple epithelia and have each cell anchored to the basement membrane, while the stratified epithelia cells, some of them touch the basement membranes and others are laying on top of each other. So these are known as stratified. So if you are simple epithelia, every single cell in the tissue will be attached to the basement membrane. But if you are stratified epithelia, they will have cells on top of each other and not all contact the basement membrane. Just remember that the basal surface of the epithelia is the surface that is facing the basement membrane, while the apical surface is the surface that faces away from the basement membrane. You see, that's simple. So simple epithelium, you see every single cell, 1, 2, 3, 4, 5, is bound to the plasma membrane, while in stratified, some cells are simple, but the others are on top of each other. And in the pseudo-stratified, you have half and half. Some of them are touching the basement membrane, and others are on top of each other. So you have some cells like here, a portion of it is touching the basement membrane and the rest is on top of the other cell. So that's pseudo-stratified. And the cell shapes, you have the sunny side up, egg, like a squamous cell shape. And this is the cuboidal, like a box shape. And that's columnar. It is like a rectangle, more long than it is wide. The simple epithelium has a single layer of cells. The simple epithelium has a single layer of cells. There are 3 types named by the shape of their cells. There are simple squamous, thin, scaly cells. There are simple cuboidal, squarish, or round cells. And there are simple columnar. These are all types of simple epithelium. So you know with simple epithelium, no matter what kind of simple epithelium are you? Are you simple squamous? Are you simple cuboidal? Are you simple columnar? In the end of the day, there's something in common, and that every single cell must be touching the basement membrane. The simple squamous are thin, scaly cells, and the simple cuboidal are squash or round cells. The simple columnar, they are tall and narrow cells. In the 4th type is the pseudostratified columnar. These are half and half. Not all the cells reach the surface, but all reach the basement membrane, and they falsely appear stratified as some cells taller than the others. These goblet cells are very important, and they are on the exam. These goblet cells, they secrete very important mucus, and they are wine glass shaped mucus secreting cells in the simple columnar and pseudostratified epithelium. There's a single row of thin cells. It permits the rapid diffusion or transport of substances. It secretes the serous fluid, and the locations are the alveoli, the glomeruli, the endothelium, and the serosa. And that's the single layer of the square or round cells, and the absorption and secretion and it's for mucus production and movement. So a very important question on the exam could be, what are goblet cells? These are mucus secreting cells. And in what type of tissue do we find goblet cells? Epithelial tissue. in the pseudostratified epithelia and the simple columnar. I'm sorry?
They're not in all of the epithelial.
No, it's just on the wine glass. These wine glass shaped mucus secreting cells, they are only found in the simple columnar and the pseudostratified epithelium. Okay? Okay, so the simple squamous epithelium. Where do we find the simple squamous epithelium? We find it looks like a single row of thin cells, and they permit the rapid diffusion or transport of substances, and they secrete the serous fluid. And the locations are the alveoli, the glomeruli, the endothelium, and the serosum. There's a single layer of square or round cells. These are the simple cuboidal epithelium. So that was the simple squamous epithelium. And you see in the simple squamous epithelial cells, you have the alveoli and the blood vessels. There is a single row of thin cells that permits the rapid diffusion or transport of substances. It secretes the serous fluid, and it's located in the alveoli and the glomeruli, the endothelium, and the serosum. And the simple cuboidal epithelium, they are like cubes. They are almost equal in their length and width. They are single layer of square or round cells. They are important for absorption and secretion and mucus production, and they are found in the liver, the thyroid, the mammary and salivary glands, the bronchioles, and the kidney tubules. The simple columnar epithelium now is an example of single row of tall, narrow cells. It has an oval nuclei in the basal half of the cell and the brush border of the microvilli. Sometimes it is ciliated, known as ciliated simple columnar epithelium, and it may possess goblet cells, and the absorption and secretion, they are important for absorption and secretion of mucus. They are found in the lining of the GIT, the uterus, the kidney, and the uterine tubes. So what happens to somebody who has gastric reflux? So they have intestinal reflux disorder. So the acid keeps brushing up, going up from the intestine through the lower esophageal sphincter. So now it's going to start the acid, the burning acid, the corrosive burning acid is going to start touching this simple columnar epithelium. So what do you think going to happen? It's going to erode it, but in order for the cell to survive, it's going to undergo something known as metaplasia. In metaplasia, in order to adapt to that stress, the same way when you lift the bicep, when you lift weight for too long, your bicep is going to get bigger to adapt to this stress. But if you stop lifting weight, it's going to go right back because it does not need to be stimulated no more. But here, when the reflux is touching, the acid is touching this simple columnar epithelium, it's going to change to stratified columnar because that's thicker to deal with this acid. Same thing with cigarette smoke. So in your respiratory tract, the cells are simple columnar, but when you smoke cigarette, this smoke is going to start touching these layers, causing the layers to undergo metaplasia. So metaplasia is when you change the cell type, for example, from simple columnar into stratified squamous epithelium to cause adaptation. But if you keep doing that, if the reflux keep going even after the metaplasia, now we're going to have anaplasia. Anaplasia is cancer. So many of the patients with gastric reflux, they don't take the medication to bring down the acidity, and the acid keep going up. Then their simple columnar is going to change to squamous, and from squamous, they're going to become cancer. People who smoke cigarette, the smoke is going to keep going into their respiratory tract, changing the simple ciliated columnar into stratified, then it's going to become cancer cell. So you're going to go have metaplasia first, adaptive metaplasia, compensatory metaplasia, and then we're going to shift into anaplasia, cancer.
that what happened to the vegetable in high blood pressure?
So in high blood pressure, what happens here is the cells didn't change. They are hypertrophied. That's a good question. Here, the cell is the same number, but they started to have more glycogen inside to get thicker and bigger to deal so they can get stronger and pump more blood. But here, the cell itself changed its nature. It didn't get thicker. It didn't hypertrophize. It didn't get thicker. It changed its structure from simple columnar into stratified squamous. Then after that, it's going to go from metaplasia into anaplasia. That's cancer. That's ciliated pseudostratified columnar epithelium. It looks multi-layered, but all the cells touch the basement membrane. These are the nuclei at several layers, and it has cilia and goblet cells, and they secrete and propel mucus. The locations are the respiratory tract and portions of the male urethra. Stratified epithelia. Stratified epithelia are these stratified epithelia, they range from 2 to 20 or more layers of cells. Some cells rest directly on others. Only the deepest layer attaches to the basement membrane. Only the deepest layer attaches to the basement membrane. So there are 3 stratified epithelia that are named for the shapes of their surface cells, known as the stratified squamous, stratified cuboidal, and stratified columnar. The 4th type is known as the urothelium and is unique to the urinary tract. It used to be called transitional epithelium. So now we're going to begin talking about the stratified epithelium. The stratified columnar are rare, and they are only in places where 2 other epithelial types meet. Stratified squamous is the most widespread, and the deepest cells are cuboidal to columnar, and they include mitotically active stem cells. The daughter cells push toward the surface and become flatter as they migrate upward. And finally, they die and flake off. This is known as exfoliation. Exfoliation is desquamation, and that is when the cells finally die and flake off, because the daughter cells push toward the surface and become flatter as they migrate upward. There are 2 kinds of stratified squamous epithelium. There are the keratinized or the cornified, and these are found on the skin surface, and they are resistant to abrasion. They are abrasion resistant. There are non-keratinized. These are found lining the internal spaces, and they lack the surface layer of the dead cells. Now let's talk about the keratinized stratified squamous, because there's 2 types. There's the keratinized cornified, and these are found in the skin, and these are abrasion resistant, while the non-keratinized, they are found lining the internal spaces, and they lack the surface layer of the dead cells. Keratinized stratified squamous epithelium, they are multiple cell layers, and the cells become flat and scaly toward the surface, and they resist the abrasion, and they retard water loss through the skin, and they resist the penetration by pathogenic organisms. Their location, the location of the keratinized stratified squamous epithelium, is the epidermis, the palms, and the soles of the feet. They are heavily keratinized. The non-keratinized stratified squamous epithelium, they are the same as keratinized epithelium without the surface layer of the dead cells, and they resist the abrasion and the penetration of the pathogens. And the locations are the tongue, the oral mucosa, the esophagus, and the vagina. Then we have stratified cuboidal epithelium. These are 2 or more cell layers. These are surface cells that are square all around. they secrete the sweat and they produce sperm, and they produce ovarian hormones, and the locations are sweat gland duct, ovarian follicles, and seminiferous tubules. This is the special type known as in the urothelium. These are multi-layered epithelium with surface cells that change from round to flat when stretched. And they allow for filling of the urinary tract with the urine that has to be deposited into the pelvis so that it can be voided out. And it is found in the ureter and the bladder. So that was all about the epithelial tissue. Now let's talk about the connective tissue. The connective tissue is the most abundant of all the 4 types of tissue, is the connective tissue. And it is widely distributed, and it is a variable type of tissue in which the cells occupy less space than the matrix. It is the most abundant, it is widely distributed, and it is a variable type of tissue in which the cells occupy less space than the matrix. So most cells are not in direct contact with each other. They are highly variable, they have highly variable vascularity, and the loose connective tissues have many blood vessels, and the cartilage has no blood vessels. So remember, the loose connective tissue is what attaches to the epithelial tissue and nourishes the epithelial tissue with oxygen and nutrients and removes metabolic waste. But the other type of the connective tissue is the cartilage, and cartilage has no blood vessels. So what are the functions of the connective tissue? The connective tissue is not like the epithelial tissue. It's there for protection. It's there for gland secretion, excretion of waste. The function of the connective tissue obviously is to connect and bind. So it is the connective tissue functions are, number 1, the binding of the organs. It connects one bone to another, muscles to bones, skin to muscle, okay, and holds the organs in place. The connective tissue is also important for support because it supports the body and its organs, and they form the internal framework of the organs. The connective tissue is also important for physical protection. It protects, it cushions delicate organs. It is also there for immune protection because the connective tissue cells attack foreign invaders. And we said that this is part of innate immunity. There are what is known as resident sentinel immune cells in the connective tissue that attack foreign invaders as a first line of defense. What are the functions of the connective tissue? The functions of the connective tissue are the movement, like bones provide a lever system for body movement, and cartilages are involved in speech. It's also important for storage. It maintains stores of fat, calcium, and phosphorus in our connective tissue. And the connective tissue is important for heat production due to the metabolism of brown fat, adipose tissue, generating heat in the connective tissue. Connective tissue is also important for transport and the blood transport gases, nutrients, waste, hormones, and blood cells. Fibrous connective tissue. Fibrous connective tissue is classified together because fibers are so conspicuous. So connective tissue is divided into fibrous connective tissue, and this fibrous connective tissue is made up of fibroblasts. We already alluded to that. We have already described what are the fibroblasts useful for. These TGF-beta cells. These TGF-beta cells are very important for depositing fiber in scar tissue formation. So They classify together because the fibers are so conspicuous components of the fibrous connective tissue. Fibroblasts produce fibers and ground substance of matrix. The macrophages arise from the monocytes. So the monocytes are circulating in the blood connective tissue. But once they detect cytokines liberated by damaged tissue or infected tissue, they resort to chemotaxis and they start propagating and moving and inching closer and closer to connective tissue. They phagocytize foreign material and activate immune system when they sense foreign matter antigen. Leukocytes or white blood cells function in immune defense, and these are found in the fibrous connective tissue. Examples would be neutrophils that attack bacteria and lymphocytes that react against the bacteria, toxins, and other foreign agents. Plasma cells arise from lymphocytes and synthesize antibodies. Plasma cells secrete heparin to inhibit the clotting and histamine to dilate the blood vessels. Just remember that the plasma cells, they are arising from lymphocytes and they are the ones who make the antibodies, while the mast cells secrete heparin to inhibit the clotting. So they are anticoagulant cells. These mast cells secreting heparin, which is an anticoagulant, and they also secrete histamine, and histamine dilate the blood vessel. So it increases blood rush to the site of the inflammation. So that's why antihistamines are very useful anti-inflammatory medications. Adipocytes are fat cells. These adipocytes are fat cells, and they are clustered in some fibrous tissue, where they dominate, and they are called the adipose tissue. What are the components of the fibrous connective tissue? Number one is obviously the fibers. There are collagenous fibers made of collagen, and these are tough, flexible, and stretch-resistant collagens. These are called white fibers due to the appearance in fresh tissue. Tendons, ligaments, and deep layer of the skin are mostly made up of collagen. They are less visible in the matrix of the cartilage and the bone. There are reticular fibers. These reticular fibers, these are thin collagen fibers coated with glycoprotein. Thin collagen fibers coated with glycoprotein. They form the framework of spleen and lymph nodes, and that part of the basement membrane are under the epithelium. Elastic fibers. Elastic fibers are thinner than the collagenous fibers, and they are made of protein known as elastin, and that allows the stretch and recoil like a rubber band. See the tendons, and these are the okay, fibrous connective tissue. The components of the fibrous connective tissue are the ground substance, which is a featureless substance surrounding the cells, usually gelatinous or rubbery. It absorbs compressive forces and contains large molecules, proteoglycans, and glycoproteins. There are glycosaminoglycans. Glycosaminoglycans are long polysaccharides composed of amino sugars and uronic acid. They regulate water and electrolyte balance of tissues, and examples would be chondroitin sulfate, heparin, and hyaluronic acid. Proteoglycans, these are found at the cell membrane. These are gigantic molecules shaped like bottle brushes. They form gravy-like colloids that hold the tissue together. What's the bottle brush?
Like bristles on the,
Bristles?
like bristles on the stick in the middle, and like you can put it inside, stuff like the bottom.
Okay, no idea what that is, but okay. Adhesive, you guys know that, right? You guys know, okay, the proteoglycans, they are like bottle brushes. Okay. Adhesive glycoproteins. These are protein carbohydrate complexes, and they bind the components of a tissue together. These are types of fibrous connective tissue. What are the types of fibrous connective tissue? There are two broad categories, the loose and the dense connective tissue. The loose connective tissue is mostly ground substance in space surrounding the cells. The types are the areolar and the reticular tissue. There are dense connective tissues. These are mostly fibers in space surrounding the cell. And these are the dense, regular, and dense, irregular cells. Areolar tissue. Areolar tissue is loosely organized fibers, abundant blood vessels, and they possess all 6 cell types and fiber types. It underlies the epithelia in serous membranes between the muscles, passageways for nerves, and blood vessels. That's the areolar tissue. The reticular tissue is a mesh of reticular fibers and fibroblasts. It forms the supportive stroma framework for the lymphatic organs. They are found in the lymph nodes, the spleen, the thymus, and the bone marrow. They are dense, regular connective tissue. They are densely packed, parallel collagen fibers, compressed fibroblast nuclei, and they are elastic tissue, forms wavy sheets in some locations, and tendons attach the muscle to the bone, and the ligaments hold the bones together. Dense, irregular connective tissue. So densely packed, randomly arranged, and collagen fibers, and few visible cells. They withstand the unpredictable stresses, and their locations are deeper layer of the skin and capsules around the organs. Adipose tissue. Adipose tissue is fat. It is tissue in which the adipocytes are the dominant cell type. The space between the adipocytes is occupied by areolar tissue, reticular tissue, and blood capillaries. Fat is the body's primary energy reservoir, and the quantity of the stored triglyceride and the number of the adipocytes are quite stable in a person. Fat is recycled continuously, and new triglycerides synthesized while old molecules hydrolyzed and released to blood. Continuing with adipose tissue. So there are two types in humans, white or yellow fat or brown fat. White adipose tissue, WAT, or white fat, or WAT, is the most abundant and significant type in adults. It provides thermal insulation. It cushions organs such as the eyeballs and the kidneys. It contributes to body contours, female breast and hip, and it secretes hormones that regulate metabolism. While the brown fat, BOT, the brown adipose tissue, or brown fat, is found mainly in fetus, infants, and children. Adults have smaller deposits of it. The color comes from blood vessels, mitochondria, and mitochondrial enzymes. It functions as heat-generating tissue. The empty-looking cells with thin margins are nucleus pressed against the cell membrane. So the energy stored insulation, cushioning, the brown fat produces the heat. Cartilage. Cartilage is a stiff connective tissue with flexible rubbery matrix. Stiff connective tissue with flexible rubbery matrix. It gives shape to the ear, the tip of the nose, and the larynx. The general features are chondroblast, cartilage cells that produce the matrix, chondrocytes. These are cartilage cells that are trapped in the lacuna. These are the cavities, and the perichondrium. Perichondrium is a sheath of dense, irregular connective tissue that surrounds the elastic and most hyaline cartilage. It contains a reserve population of chondroblasts that contribute to the cartilage growth throughout the life. Cartilage. What are the general features of cartilage? Cartilage has no blood capillaries. It's an avascular tissue. So very similar to the connective tissue, some part of the connective tissue like collagen. Collagen is a form of connective tissue, but collagen does not have a vascularization. It's avascular. So these tissues heal really slowly because they don't have any blood perfusion. The matrix that is rich in glycosaminoglycans and contains collagen fibers. The types of cartilage vary with fiber composition. Is hyaline cartilage, fibrocartilage, and elastic cartilage. Hyaline cartilage. This is clear, glassy appearance because of the fineness of collagen fibers. It eases the joint movement, holds airway open, and moves the vocal cords. That is like the growth of the juvenile long bones. The locations are articular cartilage, costal cartilage, trachea, larynx, and the fetal skeleton. Elastic cartilage. These are cartilage containing the abundance of elastic fibers. These are covered with perichondrium, provides flexible elastic support, and the locations are external ear and the epiglottis. You have a mixture between fibrous tissue and cartilage known as the fibrocartilage. The cartilage containing large, coarse bundles of collagen fibers and resists compression and absorbs the shock. And the locations are the pubic symphysis, the menisci, and the intervertebral disc. Bone. Bone is an osseous tissue. It's calcified connective tissue that composes the skeleton. So the bone is like a mountain. The bone is like a mountain because it is a reservoir. The bone is like the mountain because it is a reservoir of minerals like phosphate salt, like calcium salt, and that is important for mineral homeostasis. When calcium levels drop in the blood, hypocalcemia, we go to the bone and we initiate osteoclasts. Osteoclasts are cells that degrade. How are they able to degrade? If you are a cell and you are able to degrade, that means you need lysosomal enzymes. You need proteolytic enzymes. You need reactive oxidative species. You need metalloproteinases. These are the weapons that the cells use to degrade. So when you hear degradation, you know that these enzymes are involved. So when we need calcium in our blood, we going to activate osteoclasts that will utilize their lysosomes and lysosomal degradative proteolytic metalloproteinases and reactive oxidative species to degrade the bone and liberate bone powder and liberate calcium from the bone into the blood. And the opposite, when we have so much calcium in the blood, why is so much calcium in the blood a big problem? Hypercalcemia. Yeah, so hypercalcemia is a problem. But why is hypercalcemia a problem? Yes. Excellent. So too much calcium is going to mess up with the voltage. Remember we said that the leaky channels are going to start leaking more calcium, and then calcium is going to depolarize the cell and leading to premature contraction. And that's arrhythmia, causing arrhythmia. So that's one example. Why else is too much calcium at the end of the world? Why is it a problem? I mentioned that very first class. And that is some enzymes are off. They will get turned on with kinase. When you put a phosphate protein kinase, it's going to phosphorylate them. They're going to start working. But some enzymes, they are off, but they don't care about being activated by a kinase. They get activated by calcium. So calcium activates. So it's very important. Some enzymes, they are activated by reactive oxidative species. So the people who drink the alcohol, it gets metabolized into acetaldehyde, and then you get reactive oxidative species. That's going to turn on these enzymes that are found in the pancreas. And then they're going to cause autocatalytic degradation of the enzyme known as pancreatitis, the inflammation of the own pancreas. So some enzymes are off, get turned on by calcium, so we don't need too much of that because that's dangerous. Also, yes, any question? Somebody said something? Okay, so it's very important for calcium homeostasis, for salt homeostasis. Also, the bone is important because, like we said, it's for protection, because it's like the safe. You put inside the safe, the money protects the money inside the safe from being stolen. Your brain, your heart, and lungs, they are more precious than the money, but they are encapsulated by a safe that is this very, very important skull. And it gives us our tensile integrity, our strength to be able to move for survival. So if we want to build up bone, we utilize, remember we said earlier, blast. Erythroblast, these are the cells that would make red blood cells. Osteoblast, these are the cells who would make bone. Blast, making. Clast, destruction. The sponge bone are delicate silvers and plates that give it a spongy appearance. They are found in heads of long bones and in middle of the flat bones, such as the sternum. So, compact, dense bone is a denser calcified tissue with no visible spaces. It forms the external surfaces of all the bones. That's the compact bone. There was a question on the exam about the spongy bone also. The delicate slivers and these are delicate slivers and plates that give it a spongy appearance. It is found in the heads of the long bones and in the middle of the flat bones, such as the sternum. What are the features of the compact bone? They are arranged in cylinders called osteones. The central osteonic canal, the passage running longitudinally along the bone shaft and contains blood vessels and nerves. It is a concentric lamellae, and it is a ring-like layer of bone surrounding the central canal. There is an osteon. The osteon is a central canal and its grounding lamella. Osteocytes are mature bone cells within the lacunae between the lamella, and the canaliculi are delicate canals from the name canaliculi, radiating from each lacunae to its neighbors, allowing the osteocytes to connect to each other. The periosteum is a tough, fibrous connective tissue covering the whole bone, covering the whole entire bone. There's also compact bone. It's compact bone. Compact bone is a cylinders that surround the central osteonic canals that run longitudinally through the shafts of the long bones. And blood vessels and nerves travel through the central canal and the bone matrix deposited in concentric lamella. Blood is a fluid connective tissue that travels through the tubular blood vessels. It transports the cells and dissolved matter from place to place. It contains formed elements suspended in a liquid ground substance called the blood plasma, and the formed elements include the erythrocytes, Sometimes they're also known as the preformed element, the erythrocytes, the red blood cells, and they transport oxygen, carbon dioxide, the leukocytes, the white blood cells. They defend against the infection and disease, and these are the platelets. Okay, so what you have to know, that's very important. What you have to know about the blood, that is very important, is that structurally and functionally, when you centrifuge the blood, if you centrifuge the blood, what's going to happen to the blood? Obviously, it's going to separate based on centrifugation of the blood. It's going to separate the blood based on what? So I take a sample of blood. This is a sample of blood. I put it in a centrifuge. When I centrifuge this sample of blood, is it going to come out like that? It's not. It's going to give me a pellet in the bottom, a pellet in the bottom, and a supernatant on the top. On the bottom, you're going to have the heaviest or the least heavy. That is what the centrifuge does. It fractionizes, makes fractions. It will make 3 fractions. It will make 3 fractions. And the 3 fractions are, you have the pellet that's all the way in the bottom, and then the pellet that's all the way in the bottom, on top of it, you have, so this pellet is going to represent about 45% of my blood sample, which is 45% of my blood. 45% of my blood is erythrocytes. Erythrocytes. These are 45% of my blood. Above is about 1% or even less than 1% of my blood, and that is known as the buffy coat. Buffy coat. And this buffy coat is comprised of 2 types of cells. It is comprised of white blood cells, and platelets. White blood cells are known as leukocytes, and platelets are known as thrombocytes. Leukocytes and thrombocytes. And the buffy coat represents about 1%, while the most bottom layer, the erythrocytes, that coat is known as the hematocrit. That is known as the hematocrit. And on the highest, the topmost layer, you're going to have the rest. That's about 55%. That's the largest fraction. And that is most of our blood. And that is represented by the plasma. So the plasma is about 55% of our blood. What does the plasma have? The plasma has water, H2O, and it has proteins. It has water, it has proteins, it also has water, has proteins, it has electrolytes, it has waste, it also has hormones. So these are the 3 fractions of the blood. So the blood is concerned with all 3. It's concerned with oxygen delivery in the form of hemoglobin. So hemoglobin is carrying oxygen, delivering it to tissues, carrying carbon dioxide from the tissue, delivering it to the lungs. Myoglobin is there storing oxygen. The buffy coat has your white blood cells, so when there is a microbial infection, the proper white blood cells are going to perform the immune defense, such as your neutrophils and in addition to the neutrophils, you have your eosinophils and your basophils. And lastly, you have your thrombocytes. Thrombocytes are there to make a platelet plug, to prevent the loss of blood, to prevent hypovolemia, a drop in blood volume. And on above that, you have the largest component of your blood, that is your plasma, which is a electrolyte solution with a lot of proteins, a lot of waste, a lot of hormones, a lot of electrolytes, and that is what makes the blood polar. So these are the fractions of your blood. So the formed elements represent for me these thrombocytes and these leukocytes and thrombocytes. So which cells are involved in clotting? Leukocytes, erythrocytes, or platelets? Platelets. That's a blood smear. So erythrocytes appear as pale pink discs with light centers. And leukocytes are slightly large and have variously shaped nuclei, and platelets are small cell fragments with no. What is this? Erythrocytes appear as pale pink discs with light centers, and leukocytes are slightly larger and have variously shaped nuclei. Platelets are small cell fragments with no nuclei. Excitability. Excitability is the ability to respond to a stimuli by changing the membrane potential. So developed to highest degree in the nervous and the muscular tissue. So this is the property that defines for us the muscular tissue and the nervous tissue, where there's excitability, and that's being able to respond to an input through an effect, an output known as changing the membrane potential. We change the membrane voltage based on a stimulus that we receive. It is developed to the highest degree in the nervous and muscular tissues so that they are called excitable tissues. Excitation is founded in charge difference voltage across the membrane called the membrane potential. In nerve cells, changes in voltage result in rapid transmission of signals to other cells. In muscle cells, changes in voltage result in contraction, shortening of the cells. Nervous tissues. These are tissues specialized for the communication by electrical and chemical signal. So what's the difference between a nerve cell and a muscle cell? Well, we begin with the similarities. Both of them are excitable. But once the nerve cell is excited, it allows for the passage of information. It mediates for the transmission of information. But in muscle cells, it changes the changes in the voltage do not mediate the transmission of signals. They just cause contraction or making the cell more shorter. Nervous tissues specialized for communicating by electrical and chemical signal. Specialized for communication by electrical and chemical signal. It consists of the neurons, the nerve cells that detect the stimuli, and they respond quickly and they transmit the coded information rapidly to other cells. There are neuroglia. The neuroglia are glial cells that protect and assist the neurons and are the housekeepers of the nerve system. What are the parts of a neuron? You have, first of all, the cell body, and that houses the nucleus and the other organelles, and it controls the properties of protein synthesis. There are dendrites. Dendrites are short branched processes that receive signals from other cells and transmit the message to the cell body. Axon is the nerve fiber. It sends the outgoing signals to the other cells and can be more than a meter long. Neurons and glial cells. So these neurons, that shows you that's a neuron and that's a glial cell. These are the glial cells, and the neurons have long axons, and that's the cell body, and these are the dendrites. These are short branch processes that receive the signals from other cells and transmit the message to the cell body. While the axon or the nerve fiber sends out the outgoing signal to other cells and can be more than a meter long. Muscular tissue. Muscular tissue is specialized to contract when stimulated, exerting a physical force on the tissue, organs, and fluids. Also, it is an important source of body heat. There are three types of muscular tissue. There are the skeletal muscles, there are the cardiac muscles, and the smooth muscle. Skeletal muscle, they are made up of muscle fibers, long, thin cells. These are long, thin cells, and most skeletal muscles attach to bone, and they contain multiple nuclei adjacent to the plasma membrane, and there are striations, alternating dark and light bands. And these voluntarily, consciously, they have a conscious control over the skeletal muscle. While cardiac muscles, they are limited to the heart wall. Cardiomyocytes are short and branched with one centrally located nucleus. There are intercalated discs that join the cardiomyocytes end to end, and the striated and involuntary, they do not have conscious, they are not under conscious control. Smooth muscles, they are made of short fusiform shaped cells, and the cells have one central nucleus with no striations. They are involuntary function, and they are mostly found in the visceral muscles, making up part of the walls of the hollow organs. Cellular junctions. These are connections between the two cells, and most cells are anchored to each other or their matrix. The cells communicate with each other, they resist mechanical stress, and control what moves through the gaps between them. There are also tight junctions. These are different from the cellular junctions. The cellular junctions are connections between the two cells, and most cells are anchored to each other or their matrix. And the cells communicate with each other, resisting the mechanical stress and control what moves through the gaps between them through the cellular junction. But the tight junctions, they are like a zipper-like. They are interlocking linkage between two adjacent cells by transmembrane cell adhesion proteins. They encircle an epithelial cell near the apical surface, and they join the epithelial cell with a neighboring cell, and they seal off the intercellular space, making it difficult or impossible for the substance to pass between them. An example would be the tight junctions in the stomach and intestines that prevent digestive juices from seeping between the epithelial cells and digesting the underlying connective tissue. What are desmosomes? Desmosomes are patches that hold the cell together. They like a clothing snap. They keep the cells from pulling apart. They resist the mechanical stress, and they have hook-like J-shaped proteins that arise from the cytoskeleton. They are commonly found in the epidermis and the other epithelia. What is the difference between a desmosome and a hemo or hemidesmosome? The desmosome is a patch that holds the cells together like a clothing snap. And hemidesmosome is a half desmosome that anchors the basal cells or an epithelium to an underlying basement membrane. Epithelium cannot easily peel away from the underlying tissue. Gap junctions. Gap junctions are gap communicating junctions that channel between the cells formed by ring-like connexin. So the connexin consists of small transmembrane proteins arranged like segments of an orange around a water-filled channel. Ions, nutrients, and other small solutes pass between them. They are located in the cardiac and smooth muscle, embryonic tissue, lens, and cornea. These are the intercellular space, these are the plasma membrane, and these are the cell adhesion proteins. These are tight junctions, desmosomes, and gap junctions. Glands are also a type of epithelial tissue, and these are a cell or an organ that secrete the substance for use elsewhere in the body or releases them for elimination from the body. Usually composed of epithelial tissue in a connective tissue framework and capsule. The secretion is a product useful to the body, and an excretion is a waste product. Excretion is the waste product. Endocrine and exocrine glands. Glands are classified as exocrine or endocrine. Exocrine glands maintain their contact with the surface of the epithelium by way of a duct. So the surfaces can be external, sweat tear glands, or internal, pancreas, or the salivary glands. Endocrine glands have no ducts, but do have many blood capillaries that secrete the hormones directly into the blood. So hormones are chemical messengers that stimulate cells. So let's look at the pancreas. The pancreas is a two-in-one. It's very interesting how the pancreas is a two-in-one. It is a exocrine gland and it's an endocrine gland. The pancreas is an exocrine gland and an endocrine gland. Two-in-one. So an exocrine gland empties its content inside a duct. But an endocrine gland does not need a duct. It empties into the bloodstream. That's an endocrine gland. It empties into the blood stream. So how does the pancreas work as both an endocrine gland and an exocrine gland? It works as an exocrine gland when it secretes trypsin and chymotrypsin. So trypsin is a digestive enzyme. Trypsin and chymotrypsin. These are types of peptidases. They help in the digestion of proteins in the digestive tract. So the exocrine gland, the pancreas, would secrete this content from the pancreas to the digestive system to help in the digestion. So it's secreted through a duct. It's secreted through a canaliculi. And it secreted all of its content to help the digestion. But when the pancreas secretes insulin or glucagon, it does not secrete it through a duct. It's secreted immediately to the bloodstream. The pancreas would secrete insulin into the blood to help the uptake of glucose from the blood and lower the hyperglycemia, restore it back to euglycemia. And when I have a drop in glucose level, I have hypoglycemia, the pancreas again would secrete glucagon, and glucagon would help the removal of glucose from the liver into the blood, and that would elevate from hypoglycemia into euglycemia. But in the end, that's an example of an endocrine function because we emptied the content into the bloodstream directly. But in the exocrine gland, we don't. In the exocrine gland, we secreted trypsin and chymotrypsin into the ducts of the gastrointestinal tract. Is that point clear? Glands. These are unicellular glands, and these are secretory cells found in an epithelium that is predominantly non-secretory. And it can be exocrine or endocrine. Examples would be mucus-secreting goblet cells in the trachea or the endocrine cells of the stomach. These are the epithelial cells. That's an exocrine gland, and that's an endocrine gland. You see, the endocrine gland is associated with blood, with a blood vessel, while the exocrine gland would make a duct. It is not connected at all to a blood vessel. Arterial blood supply. You see, this is the arterial blood supply, and when the blood is passing through, that's the general structure of endocrine and exocrine gland. What's happening here is that this exocrine gland, this endocrine gland, is this an exocrine or an endocrine gland? The gland is in the back. The gland is in the back. What's in front of it? Endocrine, yes, because it's emptying into the blood. Excellent. Exocrine gland. So the general structure of a multicellular exocrine gland is it has a capsule, a connective tissue covering of exocrine gland, and a septa or a trabecula, and these are extensions of the capsule that divide the interior of the gland into large compartments called the lobes, or finer septa that may divide each lobe into microscopic lobules. The stroma are connective tissue framework of the gland, and they support and organize the glandular tissue. There is parenchyma. Parenchyma are cells that perform the task of synthesis and secretion, typically cuboidal or simple columnar epithelium. Exocrine gland structure. The exocrine glands are classified by the structure of their glands and secretory portions. The duct shape is, there are unbranched ducts known as the simple ducts, and there are branched ducts known as the compound ducts. The secretory portions are the tubular or the narrow secretory portion, and they are a duct, and the secretory portion are of a uniform diameter. While in the acini or the acinar, the secretory cells form a dilated sac, known as the acinus or the alveolus. There are tubulo-acinar, tubulo-alveolar secretory cells that are in both tubular and acinar portions. These are simple coiled tubular glands. Example would be the sweat gland. And these are compound acinar glands, such as the mammary gland. And these are compound tubulo-acinar glands, such as your pancreas. What are the types of secretions secreted by the glands? Glands are also classified by their secretions. There are some serous glands, and they produce thin, watery solutions, such as perspiration, your sweat, milk, tears, and digestive juices. There are mucous glands. Mucous glands produce glycoprotein called mucin, which absorbs water to form the mucus. And goblet cells, these are unicellular mucous glands. The mixed glands contain both serous and mucous cell types and produce a mixture of the cell types of the secretions. The exocrine glands classified according to how their secretions are released. You have their eccrine or merocrine glands. They release their products by exocytosis, and there are apocrine glands that secrete the lipid droplets covered by membrane and cytoplasm buds from the cell surface, and there are holocrine secretions, and these are cells that accumulate a product and the entire cell would disintegrate. Okay.